All right, hello everyone. Thank you very much for coming. We have a great webinar today with our very own Dr. Rio Vetter. It's going to be talking about electrode technology. Yeah, I couldn't be more excited about this if I was at home in my PJs watching this, which I'm sure a lot of you are. Future webinars. Next week, we're going to have May 14th, Customize Your Science with our own Jamie Hetke, our engineering guru. And on May 21st, we're gonna do a SmartBox Pro unboxing. We're gonna set it up just like you ordered a SmartBox. It came to your office lab, and you're gonna open it up and connect all the wires and see just how easy it is to set up and start using. So about Dr. Vetter, he is our CTO. He leads the research and development of new technologies towards innovative new products directed at research and clinical applications. Dr. Vetter has technical expertise in the field of neural interface technologies, including MEMS-based electrode technologies, electronic systems, and decoding algorithms to interface with the nervous system. His work has led to numerous scientific publications and patents. Dr. Vetter will present a broad overview of the NeuroNexus innovative MEMS-based electrode technology. Throughout his talk, he will discuss leading-edge technologies used for neurophysiology recording, stimulation, and optogenetics, used to interface with the brain, spinal cord, and peripheral nervous system. And with that, I'm gonna say, take it away, Rio.
Thanks, Matt. Let me share my screen here. Okay, good morning, everyone. Good afternoon to those of you in Europe, and good evening to those of you in Asia. We have a diverse crowd today, so I hope to touch on some topics that will resonate with the different disciplines. We've got neuroscientists and engineers and med tech folks all logging into this. So I just want to mention that this by no means is going to be a deep scientific talk, nor a deep technology talk, but rather a broad overview of our technology and our product lines and some applications. So with that, I'd like to again, thank you for taking time out of your day, and I hope you enjoy this talk. So first, introduction to NeuroNexus. We are located in Ann Arbor, Michigan is our headquarters, and we have a satellite office in Boston, Massachusetts. This is our facility here in the Technology Park of Ann Arbor. We were founded in 2004, University of Michigan spin out, and then later we were acquired by a medical device company at the time known as Great Batch. They are now referred to as Integer. And then we later on spun out and joined a spinal cord stimulation company, NeuVectra. And then in 2019, we actually spun out and became independent again. So we've been through several phases of the company, each giving, adding value to our business and our technology. And you know, it's been a fun ride so far. So we are comprised of mostly technologists, whether it's scientists, engineers, or technicians. So we're a heavily based technology company. So just a quick outline of my talk. I'm going to talk a little bit about some history of microelectrodes and how they relate to NeuroNexus and the technology that we currently offer. And then I'm going to dive a little bit deeper into the NeuroNexus technology, and then give an overview of our design space and product lines, and then finish with some interesting applications of how folks are using our electrodes. So starting in the 50s, you know, there was work prior to this. We're all familiar with the Helmholtz work and the Hodgkin and Huxley patch clamp work, but it wasn't until 1953 that the first glass pipette microelectrode was used for extracellular recording. And this was a glass pipette filled with a platinum wire, had a 10 micron tip, and you can see the extracellular recording down there. So this was a single electrode. From there, there was a big development where Hubble developed the first microwire-based electrode. So this was a big improvement because of the mechanical durability of wires compared to glass. And he was able to record from the cat dorsal root for periods up to an hour. And you can see the recording there. Later on in that decade, Stromwasser reported the first microwire bundle, and this was an array consisting of 4 wires, and he was able to record up to 7 days. If you're involved with any operant conditioning studies, you're probably familiar with the Olds work that was reported in 1965, and he used 9 nichrome wires and recorded up to 7 days with 50% yield of action potentials. Naka reported in 67 the use of tungsten microwires. These were 127 micron diameter, 1 micron tips. And here he was able to record up to 1 month. And then in 1969, there was really the beginning of a revolution as far as electrodes go. And Professor Wise, who at the time was a graduate student at Stanford University, reimagined what a neuroelectrode would look like. And this was using silicon as a substrate, using MEMS-based technologies. And so this is the first report of that in a 1969 report where he developed an electrode with silicon substrate, silicon dioxide, uses the dielectrics. And I'll talk more about this in a little bit. 1971, the NIH formed the neuroprosthesis program. And this was really a concerted effort to push the field forward. And it brought researchers together to collaborate and to disseminate new developments within the area of neural engineering, neural interfaces. In 1973, the Bach electrode was reported on. And the contribution from this electrode was it included the first flexible interconnect, as you can see in the drawing here. Schmidt then reported in 76 recordings. These were iridium wires that were coated with parylene C, and he was able to record 223 days in monkey cortex. And then it wasn't until 1988 that the first report of the Michigan probe came out. And so a little bit of history there. So Ken Wise reported in 69 the first silicon probe, but then it wasn't until he moved and established a faculty position in the early 70s at the University of Michigan, and then established NIH funding in the early 80s, that he was able to continue this probe development work. And then, as shown here in 1988, this is the first report of a silicon probe in vivo, and it really demonstrated the value that a laminar array brings. And if you look at the signals in this second column, you can see the polarity of these signals change as a function of depth with respect to the orientation and position of the neuron. Around that same time, there was a parallel effort coming out of Utah, and the Utah electrode was reported on. And this was also a silicon-based electrode, a very different manufacturing process, but still based on a silicon wafer. This was 100 micron, 100 shank electrode with 100 electrodes, each electrode at the tip of each shank. In 88 and 92, Phil Kennedy reported his cone electrode, and this was a completely different type of electrode. And this was an electrode where he seeded it with bioactive molecules, neural growth factors, with the goal of inducing neurite growth into the cone and creating a more reliable interface between the neurites and the electrode sites themselves. In 1994, the Center for Neural Communication Technologies was launched, and this was an NIH-funded center that was in place for 10 years during this phase, and this really amplified the efforts that were put into developing and refining the silicon probe technology. And really the focus was on acute probes. So these are probes that are used temporarily and then taken out of the animal or the subject. And the goal of this center was to disseminate these probes throughout the world for researchers that could use those. After that, NB Labs was a commercial entity that was offering microwire arrays for sale. And there were reports that recordings could last up to 15 months with this technology. 1996, the Utah array was commercialized and Bionics was formed to start selling those probes to researchers. In 1999, Williams reported a chronic performance, a really nice detailed chronic assessment of the electrodes, and really brought into context the electrode-electrolyte interface and the value of impedance spectroscopy to understand the failure mechanisms, both from a device or abiotic failure to biotic or the biological foreign body response. Carmos reported their array. This was out of Hungary, and this was a multi-channel array that began with an insulated tube where these wires were fed down through holes and extended out of the tube and then cut. So you have an array of wire or electrodes that extend out of the surface of the tube. In 2002, Cyberkinetics was formed, and this was really to take the Utah array and focus efforts for human brain-machine interface applications. In 2003 and 2004, we reported the first chronic performance assessment of the Michigan probe, and we were able to get reliable recordings out to 55 weeks in rats. About the same time, Miguel Nicolelis reported chronic performance of his high channel count microwire bundles. Let's see, I don't remember how many electrodes they had, but they were recording activity out 30 days, and eventually at 18 months, they still had 58 units. And then Moxon reported her MEMS-based technology, and this was a ceramic-based technology with 4 electrode contacts. And then in 2004, NeuroNexus was officially launched. And I'm going to talk more about NeuroNexus. And at this point, I'm going to pivot away from the electrode fabrication and development efforts, while also acknowledging that from 2004 onward, there were tremendous efforts, and we could talk days and days about all the different activities. And it's really been remarkable, all the different efforts that have come out. Here's just a quick snapshot. I won't go through all of these, but here's some examples of some of the other efforts. There's a transparent ECOG electrode that came out of the University of Wisconsin. Thomas Stieglitz has made tremendous contributions in the area of flexible arrays. Patrick Ruther and his group have made tremendous efforts as well. Keith Matheson with his micro LED work, and the list goes on and on. So it's kind of interesting to look at the progression of microprocessors and computational power. And so if you look back at the work that supported computational processing prior to the 50s, it used vacuum tubes, which we're all familiar with. And this example here is the ENAC computer, where they used 17,000 vacuum tubes, and it consumed 150 kilowatts of power, which is just remarkable, the resources that were required to run that computer. And then in 58, Jack Kilby put together the first integrated circuit, and this was a game changer. There was a step change in the technology. And from that point on, we're familiar with Moore's law, that the number of transistors should double every 2 years, and that's brought us to where we are now with a 2019 AMD chip that's got 39 billion transistors in a single chip. Well, if you look at this progression of technology, I would argue that neuroprobe technology follows a similar trend. And you look at the early work of discrete wires, and then with the introduction of MEMS-based electrodes, there was literally a step change that allowed for much smaller devices to be made, much higher channel count, made in batch, all these benefits. And these developments have progressed to the point where we are today. I think arguably one of the most sophisticated probes in the world right now is the Synapse probe out of Italy. And this is a probe with active circuitry integrated into it. It's a multi-shank electrode that is capable of sampling of over 1,000 channels simultaneously. And if you look at the size of the back end, which is really important from a packaging standpoint, it's only a 3 millimeter by 3.5 millimeter in size. So taking a step back real quick, what are MEMS? So MEMS, if you're not familiar, if you're not a technologist, MEMS are these microelectromechanical systems. So all of our smart devices have these in them, whether it's an accelerometer or gyroscope. But it's basically taking a device, a mechanical or electrical device, and miniaturizing it using thin film processing. So thin film or MEMS, that whole area, that technology brings a lot of favorable attributes for use in neuroelectrodes. And one of the most important things is these are made in batch. So they're made in a clean room, as you can see in the photo on the top right. Bottom right shows a photo of one of our wafers that's been fully populated with probes. So these are probes. There may be 1,000 probes on a wafer. The number varies depending on the electrode design. But the last step of this process is to release those probes from the wafer. But it really introduces an ability to precisely engineer the details of the electrodes, whether it's size, shape, materials, mechanical properties, etc. So just quickly to go through how these things are fabricated. This is just a cartoon illustration. You can see on the left here, you have a substrate. So in our case, that's a silicon wafer. You deposit a thin film. This might be a dielectric, so an insulation layer. And then you deposit photoresist. Photoresist is how you define your features on your device. So within the photoresist, you use optical lithography and create a feature within that photoresist. Photoresist is just there temporarily to create features on the device. Once you've created an opening in that photolithography layer or in that photoresist layer, then you can etch to a prescribed depth and create these features. And then it's a cycle. So you keep building up these devices. It's similar to an additive manufacturing process. And then here's an illustration just to define the different areas of a device. So this is a cross section of, let's say, a shank with 3 traces. So you have your substrate. Sometimes the substrate is part of the device. Sometimes the substrate is only a carrier for the fabrication process and then is removed later. But there's a variety of different material options for the substrate. And then you have to have an insulation layer to electrically isolate your traces from the substrate. And these could be a number of different material options. And then you have your traces, and then you have a top dielectric layer that insulates the traces from the electrolyte. And then you have your electrode surface itself. And again, this manufacturing process is amenable to many different materials, which is really opens the doors for different possibilities. So the one thing I want to point out is that a company like ours, making devices and innovating is different than an academic effort, because really our goal is to be able to manufacture these things, and we have to be able to manufacture them in a cost-effective way and in a way that is reproducible and is reliable. And so early in the design phase, we really are thinking about the design from a manufacturing standpoint. if you could design the world's most sophisticated probe, but you can't manufacture it cost effectively, then it's dead before it even begins. So it's just a different approach to the design process that we take as compared to if we were in a university lab trying to develop the next best thing. So taking it up a few notches, here are some examples of different electrode designs. This up here is a 256 channel electrode, 8 shanks with 32 electrodes along each shank. These are your bond pads on the back end. This is an example to calibrate people on the size. This is an electrode sitting on the back of a penny. This is Abraham Lincoln on the back of the penny. And again, you can make any size and shape really that you can draw on paper. This is a tetrode recording here. This is an electrode where holes or openings were created and the electrodes were moved right to the edge of the substrate. Same goes for the polymer devices. You can make a variety of different configurations. This is a long polyimide electrode that was attached to a wire for deep brain mapping studies. This is an ECOG electrode that's very common in use right now. We can also form these polymers into different shapes. This was a cylindrical form that was used for deep brain stimulation. And we can make very long interconnects as needed. So to give some illustrations of how powerful this technology can be, this is an example looking at data from a subcellular aspect. And this is a polytrode electrode where the local field potentials were recorded, and there's a common analysis technique referred to as current source density analysis. That's the second spatial derivative of your field potentials. And what that allows you to do is resolve or locate where your current sinks or sources are. So in this diagram, with respect to this pyramidal neuron, they were able to identify that the current sinks and sources were down here with respect to the electrode sites and the recordings that were collected. If you look at things from a different viewpoint, so this was a report that came out of Yuribizaki's lab and also illustrates the power of this technology. So because he knew exactly the precise location of these electrodes, he was able to triangulate the location of the neurons because he was able to record from a single cell from multiple points in space. And in doing so, he was able to determine whether or not these were pyramidal neurons or interneurons, whether they were excitatory, the red lines, or inhibitory, the blue lines, and then whether they were functionally connected, the filled symbols, or whether they were not functionally connected. And these were the empty symbols. You see some over here, they're kind of faint. But anyway, just another demonstration of the power of this technology. And then taking that up a level to more of a systems approach, this is an example of an 8-shank electrode with 8 recording sites on each shank. And here you see the corresponding data. So each trace or each color band of traces is collected from electrodes on one shank. So what you see is a lot of similarities within a shank, so within a barrel, or a column, I should say, of this cortex, but you see a lot of differences between shanks. So again, it just shows that there's a lot of information in there that can be collected with respect to the spatial arrangement of the electrodes. And then lastly, this is another famous example out of Bizaki's lab where he was able to use current source density analysis to literally identify the outlining CA1 and CA3 boundaries of the mouse hippocampus. And that again was identifying where the sinks and sources were using current source density analysis. So on that note, as I mentioned earlier, literally anything you can draw on a piece of paper, as shown here, can be designed and fabricated. This is an interesting electrode where there was holes to be opened with annular electrodes around each hole and then a site at the tip. And the idea was that this opening was going to be filled with a bioactive molecule to induce neurite growth into the array and help secure that interface. Some more interesting examples, just unique examples. These were tetrodes that were desirable, and then they also wanted a linear array and a implantable reference site, local reference. This was a sieve electrode, so it was designed to be used in a transected nerve with holes in it and electrodes so that the neurites could extend and grow through the device itself. So taking a step back and looking at our product line, as far as electrodes go, we've got just about everything you can imagine as far as penetrating, deep, shallow, surface, nerve, whether for transected nerve or cuff, going around the nerve. Optogenetics, most of our products can be used and configured to support optogenetics research. 3D electrodes, neurostim, drug delivery, chemical sensing, all these things. And then I'm not going to talk about the instrumentation on this presentation, but I will mention that we do have this SmartBox recording system. And I think it's important to understand that the whole concept behind this is really with a view of the brain and the electrode in mind. So this was a recording system designed by Electrode people. So it's really a brain-centric interface. So as you can see here, every piece of the software includes the actual probe model that is being used. So you can interact with the data in a way that is more understandable, more conceivable. You don't have to do this mental map per se. And so that's the recording side. And then there's also analysis tools as well. And then on the accessory side, again, the importance of being successful doesn't end with just the probe design. We want people to be successful all the way through from the insertion to the data acquisition. So we have many tools and instruments that also assist with insertion or handling of the device. This is a multi-probe manipulator, so it allows you to insert up to five probes with very precise angles and insertion trajectories. And then there's a motorized commutator. This is working with Alpha Omega and a very nice, elegant solution, plug and play solution. And then we have in working with other companies, we've got things like ultrasonic inserter. So this applies an ultrasonic vibration during the insertion to reduce the amount of force required during the insertion. And just as a recap here, all of these things are plug and play, which is, I think, very powerful in the sense that you can literally receive things in the mail, plug everything in, and do an experiment within hours. And we do this often. We go out to labs and we'll bring equipment or ship equipment out. And, after a short period of time for setup, we're collecting data. So just to highlight a couple of the more interesting products, the Matrix, this is our 3D electrode. So it's a truly 3D electrode where you can record, there's a recording sites along the shank, the depth of each shank, and then there's a number of different shank configurations. It goes up to 256 channels currently. And it's a modular system, so you can add ECOG arrays, or you can add multiple arrays. And it started out to be a system designed for non-human primates, but we've extended that now into medium and small animal packages. So the smallest package that could be used, this could be used on a mouse or a rat. It's literally a connector with a small PCB and then a 3D array extending out from the bottom. This is just showing a photo of a primary motor cortex and a macaque, Rhesus macaque. And there are two 128 channel arrays and two 32 channel ECOGs here. And just to illustrate, the power of this technology. I'm still waiting for somebody to report, the coherence between ECOGs and local field potentials and unit recordings. Maybe that's out there, but I haven't seen that, and that's something that I think could be very interesting. And I mentioned earlier, we provide accessories. So we feel strongly about the importance and the value of a precisely controlled insertion. So this is a computer controlled inserter that can insert in steps of 0.5 microns per step, and up to speeds up to 8 millimeters per second. So this is a matrix array being implanted. This is a vacuum interface. So once the device is inserted, the vacuum is replaced or is turned off, and then the insertion tip can be removed. In this case, you see the researcher was a little nervous the array might stick, so he had his tool there just in case. But upon removal, there was no sticking, and you can see the array is left afloat with the pulsations of the brain. We also offer EEG arrays. These have become very popular for small animals. These are a flexible grid. I won't spend too much time on this, but it opens new doors to recording data. On ECOG arrays, same thing, but these are designed to be placed on the surface of the brain or over the top of the dura, and can be made very, very small as needed. We've got a whole line of optogenetics probes. Pretty much any of our probes can be configured in a way that support optogenetics. This is an example of a 4-electrode configuration with fibers that extend down each shank. I guess I'm not showing it, but we offer up to an 8-fiber configuration. We also offer tapered fibers. For example, if you want a flat fiber, you can extend light out the tip, or if you want to extend it over a greater area, you can use a tapered fiber. Or if you want to get more sophisticated, you can use the theta station offered by or developed by Optogenetics. And this allows you, with the manipulation of this micrometer screw, you can adjust the position of the output of the light, which is also very powerful technology. And then we're also working on some micro LED probes, and this is in collaboration with the University of Strathclyde. And it's a matter of converging their micro LED technology with our probe technology. And so we're really excited about that possibility. And then we have a number of drives. This is another popular area that people are really navigating towards. And these micro drives allow you to adjust the position of the probe over time, whether it's once a week or once a month. But as your signal quality starts to degrade, you can adjust the position and you can regain some of that lost signal noise. And these drives come in a variety of different configurations. This is showing a particular drive that supports Optrodes or optogenetics probes. We have probes that are designed for deep brain mapping. This is an example of our vector probe here. These, I think, can go up to about 80 millimeters in depth, maybe even a little bit longer. And they're offered in 16, 32, 64, and now 128 channels. I'm not sure if this slide is coming through very well, but what you're looking at here is a snapshot of the neurons that were sorted as a result of this deep brain electrode. So this axis here, these numbers correspond to the electrode numbers. So electrode 20, electrode 13, 21. And then the x-axis here is the neuron, the number of neurons. So in this particular study, they were able to sort out 26 unique neurons. And what's interesting to me is if you look, in some cases that one neuron will show up on 4 or 5 or even more electrodes simultaneously, and with different forms of the wave shape. But it's really an interesting example of the type of data that you can collect with this type of probe. And then moving a little bit into deep brain stimulation, this is some of the work that we did where we took a polymer array and wrapped it around a flexible carrier, really with the goal of providing more directionality in the stimulation. So this is some modeling work that shows the different electric fields that can be generated as you activate different subsets of electrodes. So the red electrodes are the activated electrodes, and the blue electrodes would be either neutral or return electrode. And why is that valuable? So this illustration here shows just that. So in the case of DBS for Parkinson's disease, if you hit the target perfectly, the subthalamic nucleus, then you can proceed with omnidirectional stimulation. But if you miss the target slightly, you want to minimize any side effects that go into adjacent structures. So in the case of this DBS electrode, you could turn off the electrodes on one side and allow the others to activate. And this directionality aspect was validated in the non-human primate model at the Cleveland Clinic. And I won't go through all of this for the sake of time, but if you care to read more about it, the reference is down here on the bottom. So here's just a snapshot of the data that was collected out of a matrix, 2 matrix arrays. These are both 128 channel arrays implanted in the hand and arm area of primary motor cortex. And pretty standard stuff, but shows how much data can really be collected simultaneously. So I'm gonna pause this real quick so that I don't interrupt the audio while I talk. But this is an example of a subject implanted with the matrix array, and he's been trained to control a cursor on the screen by using this lever and arm motion. And what you'll notice is that we're listening, hopefully the audio comes through clearly, but we're listening to a single channel, a single neuron, and that neuron fires and has a preference for a forward motion, which is kind of interesting. But let me restart that video here. Okay, hopefully you could hear that. And then this is looking at the recording system. Okay, well, excuse me, I hope you could hear that, but anybody that collects that kind of data, it's like music to your ears when you hear neurons like that. So it's always kind of fun. Here's another example, going outside of the brain. So this is in the vagus nerve of a rat, and just showing how an electrode like ours can be inserted. So it was inserted along the length of the nerve, as you can see here. Here's the illustration to show exactly where we think the tip is below the surface. And this was a matter of about an hour later, recordings that were obtained. And you see a lot of coherence across the channels, which is to be expected given the orientation of the electrodes with respect to the fibers running through the nerve. But again, just showing that these things can record well inside nerves. So if we look at the spinal cord and using these devices in the spinal cord, this was some work to use our electrodes as a tool to help understand the stimulation waveforms that were being used during clinical spinal cord stimulation for treatment of pain. So on the left, you see these are models of a paddle electrode, and these are percutaneous electrodes. And then this just shows some modeling work showing how you can adjust the electric field in the nerve with respect to activating or deactivating different electrode contacts. So once you get that in place, then you can take an electrode like this and penetrate it into the nerve and record actual activity from the interior of the nerve. So this was a 64 by 4 electrode, so 256 channels that were inserted into the nerve or into the spinal cord. The spinal cord is here. These are our surface electrodes. So this was a polymer substrate electrode that was placed on the surface that would deliver the electric field stimulation. And then here you can see, what that would look like from a cross-sectional standpoint. And then these are the recordings within minutes after insertion. Sorry about the glare, but this was just taken with a phone. And, if I were to pause it there, but, if you look across the channels, I mean, there's just, channel after channel that shows very nice signal to noise ratio. And this is from within the swine spinal cord. Another interesting example is using these in cardiac applications. So as a little background, this is a clinical user interface for mapping during open heart surgeries or even outpatient surgery, outpatient assessments. But what they're doing here is feeding a catheter up through the interior of the heart and point by point mapping out the heart and the signals. And what they're trying to really do is understand where there are interruptions in the signal propagation, so that they can go in and ablate those areas and try to rectify, the situation. Well, this is a very tedious and coarse process. And so what we did was provided a technology that would allow for much smaller scale mapping. And so this is an electrode array that's been placed on the surface of the heart. Each black dot here is an electrode contact. And then your interconnect comes off this way and then out from the chest cavity. And one of the real challenges with this project was designing an electrode that would stretch with every contraction and expansion of the heart, because what you don't want is an electrode to slide or move with respect to the tissue, because then you get a motion artifact in your signal. And so let me just show you the video of this. So hopefully that also came through okay. And then here are the EKG signals recorded with our recording system, the SmartBox. And if that wasn't challenging enough, this was taking that down to the mouse model. So this is a mouse heart. Sorry for the poor quality. Again, this was just taken with my phone. But this opening here to calibrate you is about 3 millimeters in diameter. You can see an Omnetics connector here for scale purposes, for reference purposes. But the mouse heartbeat averages about 500 beats a minute. So you can imagine, how fast things are going. And then, we were using our recording system to also collect that data. And there's an electrode array sitting right here on the surface of the heart, and it collected, very nice signals. Okay, and then finishing up, I just want to make the point that, we really are a team of technologists. The founding team and the management team are a team of scientists. And so we are constantly working with researchers out in the field, and we're responding to different technology asks from researchers. And then we're out interacting with people and trying to understand where there are unmet needs and where we can innovate and contribute further to the field. So, we encourage people to reach out to us. We think of ourselves as solution providers, and we're constantly, every week talking with researchers about coming up with a solution to their particular problem. And with that, I'll just finish with this slide. One of my favorites. This is another Bazaki image, and this shows his electrode. 8 shanks with 32 electrodes on each shank. So what you're looking at here is an array of signals that were recorded and mapped over the hippocampus. So you have 32 traces by 8 across.
All right. With that, I'm going to say thank you very much for joining. Thank you very much to Rio for your time and putting together this great presentation for us. And that's it. Thanks, everybody.
Thank you, everyone.